I'm Chase, and I have some stuff to say about mitosis. It's not your mitosis, it's mitosis! Ta-da! Mitosis is the dividing stages of the cell. Now, it's made up of 4 stages: prophase, metaphase, anaphase, and telophase. It's simple. Listen, you can remember it with this easy acronym. Please marry Anna tomorrow. Please. She needs it.
There's another step in this process. You already learned about interphase, which occurs before mitosis, but basically the cell is getting ready to split, but it continues its normal cell functions. Now let's begin with prophase.
Now in prophase, that's when the cell gets a signal it's time to divide. So what happens is the cell is preparing itself by replicating the DNA, just getting the centrioles in the right position, just anything required for mitotic division. So the cells are condensing the chromosomes, which is twisting all around the nucleus, and the nucleus also disappears. So now what's happening is the centrioles move to the opposite sides of the cell. However, in plant cells, because they don't have centrioles, they use these organelles called centrosomes. So yeah, centrosomes. And those are located in the cytoplasm near the nucleus. Now once this happens, the mitotic spindle forms. These are spindle fibers made up of microtubules, which grow from each centrosome or centriole, depending on how it is. Between prophase and metaphase, there's this in-between stage called prometaphase. Convenient, right?
First, in prometaphase, the nuclear membrane breaks down. The microtubules connect the kinetochore of each sister chromatid and the arms of the chromosomes to the spindle poles around the centriole. The kinetochore is a protein structure that forms at the centromere of each chromatid.
Now let's talk about metaphase. Now the gist of what's happening is the chromosomes are lining up in the middle of the cell, which is the metaphase plate, perpendicular to the poles ready to be pulled apart. Now this part is important because this organization helps ensure that the new nuclei of the new cells will each get a copy of the chromosomes when separating the next phase, which is anaphase.
Anaphase begins when the enzymes break down the centromeres of the sister chromatids. The centromeres are pulled apart and separated between the two. Based on the location of the centromeres, a J or V shape appears during the division. Then, the chromatids are sent to different sides of the cell. Now the chromatids are complete chromosomes.
Telophase is the last stage of mitosis where cell division finishes up. The chromosomes assemble at opposite ends of the cell at the poles. Separation then happens afterwards. Nuclear membranes form around these chromosomes, the opposite of prophase. As this is happening, the chromosomes are uncoiling, which is making them less visible.
The spindle fibers also disperse. The cell membrane pinches in, forming 2 daughter cells, the beginning of cytokinesis. This completes chromosome separation, so you end up with 2 separate cells, each with half of the original cell.
We have reached the stage where the cell divides in 2. This is called cytokinesis. All the while the cells dividing in 2, our handy-dandy plasma membranes constrict between the nuclei. Now plants and animals have two different ways of getting this done. Plants, because of their cell walls, they have these vesicles containing cellulose, and they fuse together forming the plasma membranes of 2 separate cells. So the result is these 2 cells, and this happens during telophase. Now, unless you are a giant leaf sitting at a computer, which I hope you're not, you might want to know how animals get their business done. Animals, their plasma membranes pinch, forming a cleavage furrow until they are 2 separate cells, and this begins during anaphase.